Hi everybody, this is Mrs. Kraus, and this is our second video for the Unit 7 Lesson 3 notes on protein synthesis and mutations. The first video covered protein synthesis and the second video covers mutations. Again, this is very optional for you to view. We're not requiring you to do any work at this time, but for those of you who would like to tune in, we're here. Okay. So a mutation is going to be any change in the sequence of nitrogenous bases, so your adenines, thymines, cytosines, and guanines in the DNA code. Remember, the sequence of bases in DNA determines the sequence of bases in mRNA, which then determines the sequence of amino acids within a polypeptide. So if you change the DNA sequence, that may change the amino acid sequence as well. For example, there is a mutation in the gene that codes for hemoglobin protein in red blood cells that can lead to a condition called sickle cell disease. Now, if you look at the normal DNA here, normally we have a GAG on this DNA strand that goes with a CTC on this DNA strand, and we're using the bottom DNA strand as the template for creating our mRNA. So complementary to that bottom strand would be GAG. And then if you look at a codon chart, that codes for the amino acid GLU. Okay. However, with the sickle cell mutation, we have a T instead of an A on this top DNA strand, which means we're going to have an A instead of a T on the bottom one. Our codon on the mRNA will now be GUG. And we're going to end up with a different amino acid in the protein. So we have valine instead of glutamic acid. Okay. Now, the issue with that is that this single amino acid difference causes the hemoglobin protein to clump together, and the red blood cells actually have a different shape as a result. So normally, red blood cells look like this. They almost look like little donuts or inner tubes. And they can fit really nicely through blood vessels. But this is what the red blood cells look like when we have this mutation. It's almost like a crescent moon shape or a sickle shape. And there's a couple issues with that. One, they're not good at carrying oxygen. Okay. Two, they tend to accumulate and get stuck in blood vessels, which can cause a variety of issues. If it happens in your coronary arteries, it could lead to a heart attack. In your legs, it could lead to a deep vein thrombosis, or in the brain, it could lead to a stroke. Another example of a DNA mutation that causes a genetic disorder in humans is Tay-Sachs disease. And this is a gene that codes for an enzyme. It's normally present in the cell part called the lysosomes. And the lysosomes contain a variety of different digestive enzymes that break down other molecules. But in this case, that particular enzyme changes shape and it's no longer functional. The role of that enzyme was to break down a fat called GM2, which is located in the brain and spinal tissue. And if the enzyme cannot break down that fat, it will accumulate and push against the neurons in the brain and spinal cord. That's going to seriously impair nerve function, and at this point, there is no cure for it, and unfortunately, children who have this condition typically don't make it past age 4. Okay. Now we're going to look at the different categories of mutations, the first being gene mutations, and that either affects just one base or a short sequence of bases within a single gene. And we can further classify gene mutations into point mutations and frameshift mutations. Point mutations happen when you exchange one base for another, which is called a substitution. An example of that is the sickle cell mutation that we just looked at. Frameshift mutations are not when you substitute a base. Frameshift mutations happen when you either add in extra bases or remove bases from the sequence. Okay. Within our point mutations, which are the ones where we're substituting a base, We have three different categories of point mutations based on the effect that those gene mutations have on the resulting amino acid sequence. Now, a silent mutation is when the DNA change results in an mRNA codon that actually gives you the exact same amino acid. Remember, there are multiple codons that can code for the same amino acid. If we even look at, let's see, our codon chart here, we see that both UAU and UAC code for the amino acid tyrosine. So if changing the DNA changes our mRNA from UAU to UAC, that's going to be a silent mutation because it won't affect the amino acid that's put into the polypeptide. Same with if you've got a DNA change that results in your mRNA going from UCU to UCC, you're still going to have the amino acid serine. That's called redundancy in the genetic code, and again, that's a silent mutation because it will not affect the polypeptide that's added, or the amino acid that's added to the polypeptide chain. Now, a missense mutation is when changing the DNA changes the mRNA codon and you end up with a different amino acid, okay? And then the last type is a nonsense mutation where changing the DNA ends up changing your mRNA to a stop codon, which remember terminates the polypeptide. So this will end the polypeptide early, and especially if this happens at the beginning of the sequence, that could be really bad because you could have a very short truncated protein. Let's see how long I've been going here. Open main window. Okay, I've been going for 7 minutes. I have 15 minutes. That's all YouTube will allow. So let's do this. All right, so here's an example of a point mutation. So up on the top, we've got our normal DNA sequence. I use that to find my mRNA sequence by complementary base pairing, remembering that I don't have thymines on mRNA. I have uracils instead. And then I used my codon chart to find the amino acids that would go with these mRNA codons. Notice I divided it up into groups of 3 because that's how we read a codon chart. All right, now let's say I have a substitution of a base. So what happened here is where we normally had a G, we substituted a T instead. That changed the mRNA base to an adenine. And when we looked at the codon chart, ACC then coded for the amino acid threonine, which is different from the original amino acid proline. So we would consider this a missense point mutation because it changed one amino acid. Now, if it didn't change the amino acid at all, that would be a silent mutation. If it coded for an early stop codon, that would be a nonsense mutation. Okay. Now, a frameshift mutation, again, is not where there's a substitution of a base, but where we add or delete bases to or from the sequence. And that is going to result in your groups of 3 bases in the mRNA changing. So we're literally shifting the reading frame of the mRNA. For example, let's say this sentence right here is supposed to represent bases within an mRNA sequence. Notice that they're grouped in 3s, just like the bases in mRNA are grouped in codons. And let's say we deleted one letter or one base from the beginning of the sequence. But then we still grouped everything in 3s. The problem with that is now we've shifted the reading frame and our sentence no longer makes sense. This is what's going to happen in a polypeptide. Every codon will be different, and pretty much every amino acid after the mutation will be different as well. The exception to this is if you have an insertion or deletion of multiples of 3 bases. So 3 bases, 6 bases, 9 bases, because it doesn't actually change the reading frame at all. So here's an example of a frameshift mutation. So here we've got our normal DNA, mRNA, and amino acid sequence again. But let's say we added in a cytosine base before this sequence of 3 guanines. Notice how it changes every single DNA triplet and every mRNA codon after that mutation. And we end up with an extra base just kind of hanging off the end that won't code for an amino acid. So notice after our mutation, all of our amino acids are now different. We had proline, lysine, and glycine in the original sequence. Now we've got alanine, glutamic acid, and arginine. Okay, so this would be a frameshift mutation because it's shifting our reading frame, and it's an insertion because we're adding bases. Okay. Now the other large category of mutations other than gene mutations that affect one base or a few bases within a single gene would be our large chromosomal mutations that affect a big chunk of a chromosome. So here we see a duplication where we've got a large section of the chromosome that gets copied and then stuck in next to the original sequence. An inversion is where we have a chunk of the chromosome that gets flipped around and stuck back in. An insertion is when we have a chunk of one chromosome that gets stuck into another chromosome. A translocation is basically the same except it's an exchange. This green one gives a chunk to the red one, and the red one gives a chunk to the green one. And then our last type is a deletion, where we have a large section of the chromosome that gets deleted or removed. Okay. Now we've talked about some causes of mutations within our DNA replication notes, which was lesson 2. If DNA polymerase, the enzyme that's responsible for actually copying the DNA, makes an error, that can cause a mutation. Also, at any point in the cell cycle, external factors called mutagens can alter the DNA base sequence and damage the DNA. Like for example, chemicals or ultraviolet radiation would be considered mutagens. And if those mutagens cause a mutation that results in overdivision of cells that accumulate to form a tumor, we would consider those mutagens to be carcinogens or cancer-causing agents. An example would be cigarette smoke. It's both a mutagen because it mutates the DNA and a carcinogen because it mutates the DNA in such a way that it leads to tumor formation. Okay. Now, not all mutations that happen in a parent will be passed down to their offspring. Somatic mutations happen in regular body cells of the parent, so they can't be sent down to the offspring. For example, this is a mutation in red delicious apples that results in like half the apple being yellow and half the apple being red, but it doesn't get passed on to the baby apple trees that come from the seeds because it's a somatic mutation. Okay. In contrast, a germline mutation happens in the actual sex cells, the gametes, so they do get passed down to the offspring. Because remember, the sex cells are the only ones from the parent that are used to make the offspring. Okay. Now, when we hear the term mutation, we typically think, oh, mutations are bad. It's not a good thing. And oftentimes that is the case. However, there are some mutations that are considered neutral or potentially positive. For example, the silent mutations, which are one of our types of point mutations, they don't change our amino acid sequence at all. So they don't affect the polypeptide and they don't affect the organism. Positive mutations, we could end up with a protein that gives the organism a trait that's actually beneficial. For example, there are some bacteria that have a mutation that makes them resistant to antibiotics, which are drugs that would normally kill them off. So they're making a protein that somehow allows them to survive in the presence of that antibiotic, which is good for the bacterium, not great for their human hosts, but good for the bacterium. Okay, and that's it for the mutations part of the notes. So this is the second and final video for the Unit 7 Lesson 3 notes. Thank you for tuning in.